Okay everyone, in this video we are going to look at gene interaction. And what do we mean by that? Well, so far in the course, when we were looking at more than one gene, say 2 genes, we were also looking at 2 characters. For example, we looked at seed shape in P and seed color in P. Now, seed shape was determined by one gene, and we had 2 different alleles for this gene. So we have homozygous dominant, heterozygous, homozygous recessive. Seed color, what do we do? Here we go. Also determined by, also involved 2 different alleles for the seed color gene. So homozygous dominant, heterozygous, homozygous recessive. The point is we have 2 different genes, each gene determining a different character. So now with gene interaction, we're looking at more than one gene. Let's say at most we will look at 2 genes in this introductory genetics course. So more than one gene, in this case 2 genes, determining or influencing one character. And gene interaction is very important. Most traits or complex traits involve the interactions of many different genes. For example, congenital deafness in humans is caused by mutation in one of 50 different genes. So we need at least 50 genes to be working perfectly, or almost perfectly, in order for humans to hear. So gene interaction, so 50 genes interacting together to make one complex trait. So gene interaction is really important, and we're going to take a look at how 2 genes combine to influence one character as an introduction to gene interaction. So before we start this, it is important to be able, or to understand this sort of shortcut for determining genotypes, okay? So now this shortcut is based on things we've already covered. So we've seen crosses such as this, heterozygous for 2 pairs of genes. And in our previous looks at this type of cross, it has always led to, in cases of complete dominance and recessiveness, a phenotypic ratio of 9 to 3 to 3 to 1, right? Where 9, we're dominant for one character and dominant for the other character. Let's say this is character 1 and this is character 2. So 9 out of 16 progeny were dominant for both. 3 were dominant for one and recessive for the other. 3 were recessive for one and dominant for the other. And 1 out of the 16 were recessive for both. And we can symbolize the genotypes. So if this is the phenotypic ratio, we can come up with a notation system to sort of have a shorthand, I guess, summary of the genotypes if we don't care about distinguishing, say, between homozygous dominant and heterozygous. So if we don't care about distinguishing between homozygous dominant and heterozygous genotypes, we can also see a 9:3:3:1 genotypic ratio in this sort of cross right here. And it would be 9 out of the 16 would have at least one dominant A allele and one dominant B allele. So this little underscore here, what that means is we don't care if there is a big A or little a here. We're only interested in the fact that 9 out of the 16 will have at least one big A allele and one big B allele. Again, same thing over here. We don't care if there's a second big B allele or if this is a little b. We don't care about that. That's why we're putting that underscore there. So these three right here, we can symbolize like that. So 3 out of the 16 will have at least one big A allele and 2 little B alleles. The other 3 out of 16 will have 2 small A alleles and at least one big B allele, and 1 will have 2 little A's and 2 little B's. Now I can show you how we can use the forked line method to come up with this genotypic ratio right here. So all we need to do is take the cross and then look at one pair of alleles at a time. And we could do the Punnett Square, but we probably should have this memorized by now, that if we cross heterozygous big A small a to heterozygous big A small a, we will have 1/4 homozygous big A, 2/4 heterozygous, and 1/4 2 little a's. And we can summarize this as 3/4 big A underscore, because that accounts for both the heterozygous and the homozygous dominant genotype. And then this one right here will be 2 little a's. And I could diagram this out just like I did over here, but we should be able to see that 3/4 of the offspring from this cross right here will have a large B allele, and then we don't care of the other one. The other one could be big B or small b. At least 3/4 of them will have at least one big B, and 1/4 2 little b's. And then we combine these with the forked line method. So of the 3/4 that have at least one A allele, 3/4 of those will have at least one B, 1/4 will have 2 little b's. Of the 1/4 that are homozygous little a, 3/4 will have at least one big B, 1/4 will have 2 little b's. And if we multiply these together, I'm running out of space here, so I won't diagram it, I won't draw it out for you, but I'll point up here. 3/4 A underscore times 3/4 B underscore equals 9/16 A underscore B underscore. 3/4 A underscore times 1/4 homozygous B, little b here, would be 3/16 A underscore 2 little b's, and so on and so forth. So that's how we derive this genotypic ratio. And this will be very useful when we're looking at gene interaction, and you're about to see why that is the case. So our first example of gene interaction is going to be coat color in mice again. So we won't look at the A superscript Y allele that is dominant to the A allele and causes the yellow fur color and it's lethal in the homozygous condition. We're not looking at that one. But we are going to look at this gene interaction diagram, and it's going to look something like this. So we're going to start with a precursor molecule, which let's call this a white pigment or albino, or a colorless pigment. So this white pigment is going to be turned into a black pigment, which is going to lead to a black fur color, by a gene or an enzyme encoded by gene B. Now the black pigment will be acted upon to give the agouti pattern by another enzyme, and that enzyme is encoded by gene A. So in order for a mouse to have a black fur color, it needs to have at least one large B allele for gene B. In order to have the agouti pattern, it needs to have at least one big A allele for gene A. So if we were to do a cross, so let's say the P1 generation has this genotype, homozygous big A, homozygous big B, and the other parent is homozygous little a, homozygous little b. We know that the offspring will all have this genotype, or we should know by now that the offspring will have this genotype from the earlier parts of the course. And we also know in the F2 that we will have this 9:3:3:1 genotypic ratio that looks like this. Okay, so now looking at the gene interaction diagram, we should be able to predict the phenotypes of all of these mice with these genotypes. So this one right here, because it has at least one big A allele and one big B allele, it should have the agouti pattern. So the big B allele allows it to make the black pigment. The presence of a big A allele allows it to take the black and make the agouti pattern. This one right here has neither A nor B, so it's stuck with the white fur color. This guy, these offspring right here, the F1, they all have at least one big A and one big B, so they are all agouti. And then in the F2 generation, well, let's take a look. 9 out of 16 have a big A and a big B, so these are all agouti. 1 of the 16 homozygous recessives for both genes are going to be white. Now, let's take a look at this one right here. 3 have 2 little a's and at least one big B. Okay, so they can make the black pigment, but they can't put the agouti pattern on top of the black pigment. So they are just going to have black fur color. However, this one right here, this has the big A allele, but it doesn't have a big B allele. So these guys right here, even though they have this, they can't make the black pigment, so they're stuck way back here. And these will be white. Now this leads to a 9 agouti to 3 black to 4 white ratio. Okay, so 9 to 3 to 4. It's kind of strange. And we go over this example because it is our example of recessive epistasis. Now, in a broad sense, epistasis means refers to some sort of gene interaction or interacting genes. Specifically, it means there's some sort of suppressive effect of one gene on another interacting gene. And in this case, recessive epistasis, what's happening is that the presence of recessive alleles at gene B is masking the presence of functional alleles at gene A. So the recessive alleles at gene B are masking the genotype at gene A. So they're masking the identity and the function of the alleles at gene A. Now can we see that? Because even though there are functional alleles here, and these mice should be able to make the agouti pattern, they can't because they have recessive alleles at gene B. They need the black pigment made before these guys can function. So that's an example of recessive epistasis. Recessive alleles at one gene masking the function and identity of alleles at another gene, at an interacting gene. Okay, so that's our example of recessive epistasis. And see how easy it was to come up with this ratio because we had this memorized, right? We had this sort of genotype, 9331 genotype ratio memorized. Again, you don't need to memorize interaction diagrams. You will be given those on an exam and a quiz. On exams and quizzes. So now that we have covered recessive epistasis, let's look at dominant epistasis. So epistasis, when you see epistasis, you should know we're talking about genes that interact together to influence one character. Epistasis. You should just know right away, okay, we're dealing with interacting genes. When someone says interacting genes, we mean they're interacting to influence one character. So dominant epistasis, let's look at that. Summer squash. Now, this is a neat example because here we have a white precursor molecule, and we have a gene, gene A, which is responsible for turning the white precursor into a green pigment to give us a green summer squash. And then gene B, well, it's responsible for turning the green pigment into a yellow pigment. To give us yellow summer squash. Now, it's a little different than the last. I mean, in addition to these being plants and the other example being mammals or mice. So here it's the recessive condition that is needed to go from the white summer squash to the green summer squash. And here we need at least one dominant allele to go from the green summer squash to the yellow summer squash. So for example, let's take this cross right here. 2 little a's, 2 big b's, cross 2 big a's, 2 little b's. The F1, heterozygous. The F2, we already memorized this. So 9, 3, 3, 1. Now we can go to our gene interaction diagram and predict all the phenotypes. We have 2 little a's here. Okay, so these guys right here can go from white to green, and we have at least 1 B here, so these are all yellow. Here we have 2 big A's, so okay, they're going to be stuck at white. Doesn't matter what they have here. These right here, they all have 1 big A, so we said if you have 1 big A, if you don't have 2 little a's, you can't go from white to green. So 1 big A means, sorry, stuck at white. Doesn't matter what's here. So 9 have they have 9 out of 16 of the F2s have at least 1 big A, so stuck at white. 3 of these guys, doesn't matter what's at B, they're also stuck at white. 3 little a's, okay, so these can go to green, and they have 3 little a's that go to green, and they have at least 1 big B, so 3 can be yellow, 3 are yellow, and 1 out of the 16, they can go to green, but they can't go to yellow, so they will be green. So we have a 12 white, 3 yellow, 1 green ratio. So a 12 to 3 to 1. And again, so why is this an example of dominant epistasis? So this is an example of dominant epistasis because when there's a dominant allele here, it masks the identity and the function of the gene over here. For example, let's see, okay, this guy right here. These 9 out of 16 have a big A here. It doesn't matter that they have a big B. They could have 2 little b's, 2 big B's, a big B and a little b. It doesn't matter that all of them have the ability to turn the green into yellow. We can't really tell. We couldn't tell just by looking at the white, the white color of the squash, what is here, whether it has a big B or a little b or not, because the presence of at least one A prevents us from knowing for sure whether a squash has a big B allele over here. So dominant epistasis occurs when a dominant allele at one gene masks the identity and function of another gene, or an allele for another gene.